Hello, my name is Michelle Morand, and I'm a precision cancer medicine educator and an advocate, and I am here with the cancer guy, Alexander Rolland, and co-founder of Cancer Treatment Options and Management and Liquid Biopsy Labs. And he is here to help make a video essentially about something that is a real stressor for many cancer patients, which is how to tell if they've had decent genetic testing. And specifically, we're going to talk today about tumor DNA sequencing, why it matters, how it works, how it affects your treatment. And then we're going to talk about how to get it or make sure that you've actually had good tumor DNA sequencing. Alex.
Hello. Can we also talk about my new glasses?
Sure, we can talk about your new glasses. They're very nice.
$39 at the local pharmacy.
Well, there you go. You are frugal.
Yes, so I'd like to talk about tumor DNA sequencing and how important it is in cancer treatment. As you know, it is really the fundamental, first, most important thing of precision oncology, and it's guided precision oncology for many, many years.
Okay, hold on a second. You say, as you know, how would anybody but you, who has been driving the bus here or leading the charge, how would anybody know that?
Well, I think most people are fully aware of what DNA sequencing is now. In fact, I have all kinds of people coming up to me and asking me about DNA, including doctors. The problem is everyone knows about it, but not a lot of people really know about it, if you understand what I mean.
Yes, that's right. So we might understand that it exists, and maybe we even have a sense that it's important for our
Yes.
cancer care, or it will help our doctor inform our care.
So let us clarify. Tumor
Yes.
DNA sequencing is not at all the same as germline, aka inherited mutational testing. I like to call tumor DNA sequencing as tumor DNA sequencing. A lot of people will refer to it as genetics, but I think traditional genetics really focuses on genes that are inherited, germline inherited, meaning they are passed on from generation to generation by one parent, and typically through a Mendelian inheritance pattern. That means you either have a dominant gene or a recessive gene or a combination. But tumor DNA sequencing is very different. For starters, the difference between tumor DNA sequencing and standard genetic testing is tumor DNA sequencing does not look at inherited mutations. I mean, it looks at mutations that are in the tumor that are different from the genes or from the mutations that are in our body. So when a tumor first develops, it's based on certain key mutations that occur. And that's well established, the types of mutations that occur and how many, etc. But germline inherited testing doesn't look at that. It looks at these mutations that are inherited. And the classic example is the BRCA1 and 2. And these mutations typically will be in every cell in the body if you carry it. So if you carry a BRCA1 or 2 mutation, it's going to be in every single cell in the body, not just in the tumor cells. So the takeaway is that, typically there's about 150 genes or so that we look for in inherited germline testing. These genes typically are tumor DNA repair genes. In other words, they're genes that will prevent cancers, what we call tumor suppressor genes. And they will protect your genome from, mutations and from damage, and protect the DNA from damage. And so if you have a damaged gene, such as a BRCA1, then that means your cells could develop or can start developing an accumulation of mutations. And those mutations can lead to cancer at some point in your life. And so it's important to understand that while these genes are inherited and increase your risk of cancer, they are not really targetable in most cases, number one. And then number two, they don't actually guarantee you're going to get cancer. They just increase your risk for cancers in certain contexts.
Right. So as we've talked about in other videos, if you have a mutation on both strands, one from mom and from dad, then that's often where we get childhood cancers from.
Yes, exactly.
Yeah. But if
So
we're
you
later
have,
in life, oh, sorry, please go ahead.
Yeah, you have one mutation that you're born with, and then another one just happens over time in the other copy of the gene.
Mhm.
If typically for most tumor suppressor genes, if you have mutations in both copies, then you probably won't make it through embryogenesis. In other words, you won't get born.
Oh,
Yeah, yeah. There's a lot of genes that if you have both mutated copies, or both copies are damaged or mutated, then the embryo just self-destructs.
oh, interesting. Hmm.
Yeah.
Okay, so we've got that. So right now we're up to the point where we've got inherited or germline mutations testing.
Mhm.
One of the better tests out there right now looks at 153 known inherited
Yeah,
mutations
and that's a
specific.
lot of genes. Yeah, yeah,
Yeah, so I just want to make that point because sometimes folks are tested for maybe BRCA1 and 2, and they're told they don't have any inherited mutations. I just want you to know you can test for over 150 of those. There are a lot more than just a couple right now. And again, as we get into tumor DNA sequencing, you'll see how many more mutations that matter can be tested for.
yes, correct. So tumor DNA sequencing is much different. It's just the tumor. You're only interested in the mutations in the tumor, and you want to actually compare those mutations to compare the genes that are mutated in the tumor to the genes that are in your rest of your body cells.
Mm.
So cancer typically develops, it takes about 4 mutations to turn a normal cell into a cancerous cell. Um, and these are well-defined classes of genes. They're different genes in every person, but they have similar functions. So when I say 4 mutated genes, I mean, 4 separate classes of genes. And each class could have many different genes in it. There's thousands and thousands of mutations. We have about 20,000 genes, 22,000 genes, maybe 24,000. The numbers jump back and forth, as to what's actually a gene. A gene's typically classified as having control regions and exons and introns, but we're kind of broadening our idea of what a gene is. And we're starting to look at different things like transcribed DNA and so on and calling them genes. But roughly, we have about 22,000 as of today. Genes are basically strings of amino acids. You typically have 3 different DNA base pairs that code for one amino acid. The mutations occur that change a specific amino acid. So typically, the mutations we're looking at change an amino acid in a gene. And what that does is it changes the function of the gene. So the goal of tumor DNA sequencing is typically to identify DNA base pairs that have been mutated that result in an alternate amino acid. So if they don't change the amino acid, then we're not as interested, although that's a different subject and we'll have to cover that in a different set of videos because that could definitely change the way genes function. But we're looking at the coding region in the exons. The amino acid coding regions are in the exons of the gene. So basically, tumor DNA sequencing helps to identify which amino acids have been changed and how they are now involved in cancer. So a little summary on types of genes. Typically, we like to look at genes as oncogenes and tumor suppressors. So amino acid changes can result in a gene being turned on permanently, what we call constitutive activation, or turned off permanently, which we call inactivation. So we have 2 general classes of genes, what we call oncogenes and tumor suppressors. So an oncogene is a gene that's supposed to be turned on just briefly, typically to repair damaged tissue, to replace old tissues. Most oncogenes will tell the cell to reproduce or are involved in some sort of reproduction cycle, whereas tumor suppressors are involved in repairing damaged DNA and often in preventing cells that are mutated from actually turning into cancer cells. So they don't just fix the DNA, they check and see if there's DNA here or mutated DNA. And if there's some mutated DNA, then they'll turn on what's called checkpoint genes that prevent the cell from going through its mitosis cycle.
Mm.
So typically, oncogenes are overactivated in cancers. In other words, they're turned on much more than they're supposed to be. And tumor suppressors are inactivated. So tumor suppressors are normally turned on all the time. Oncogenes are just turned on briefly to reproduce new tissue. And when they get damaged, they stay on and tumor suppressors get turned off. So it's like having the gas pedal on a car stuck on and combining that with the brake pedal braking and not working.
So if we come back to what we were saying about the inherited mutations earlier, and we were talking about maybe, you might be born with one strand of the DNA having a particular mutation in a tumor suppressor gene.
Yeah.
And then as you go through life and you experience environmental toxins and various stressors or other who knows what, and something happens at that perfect storm of those 4 mutations, if one of those has something to do with this other side of the tumor suppressor gene, is
Yeah.
that
Yeah,
that
exactly.
becomes a
Yeah,
problem, I would think.
exactly.
So
Because
we're more,
then, so if you have 2 genes, you have some functional redundancy. If you damage one, you could still go with that. But if you damage both versions of it, then that's going to turn into a cancer pretty quickly.
yeah. Oh, I
Okay.
also feel compelled to say, sorry, we talk about chemotherapy and we talk about how chemotherapy works because it kills all rapidly reproducing cells, or as many as
Yeah.
it can get its hands on, whether they're cancer cells or not. And if we go back to what you were saying, or just showing about the oncogenes, then essentially you've got these stem cells, or they're supposed to be rapidly reproducing, but they're now becoming problematic. And is that right?
Sort of.
Yeah.
So the stem cells produce the bulk of the tumor, and then they turn themselves off. It's pretty well recognized in this day and age that stem cells are the cause of cancers, and that only the stem cells that produce the bulk of the tumor can actually metastasize. And typically a stem cell is roughly is going to produce about 6 million tumor cells. So chemotherapy will kill a lot of the bulk of the tumor, but it doesn't affect the stem cells.
Right.
And that's part of the problem.
Yeah. Yeah.
And that's because the way most chemotherapy drugs work is they interfere with the reproduction cycle of a cell. So when a cell's copying itself, it will damage the DNA so that the cell can't reproduce itself. Whereas with stem cells only are active for a short period of time. They'll get turned on briefly, they'll produce a massive amount of cells, and then those cells will go on to get differentiated into the type of tissue they're supposed to be. So often tumor cells or tumors are created by stem cells that only get turned on once in a while. And so if you don't have this chemotherapy drug when that stem cell is actually producing the bulk of the tumor, it's not going to affect the stem cell.
Oh, I see.
Yeah. Yeah. So tumor DNA sequencing, is really, really important. Mostly because it tells us which targeted therapies can benefit us. And so targeted therapies have really been the crux of precision oncology for a long, long time. However, certain mutations can also tell us, what standard chemotherapy genes will work, whether radiation works, or it will be really effective or not. But all in all, it's super important to know what DNA is mutated in your cancer. And now we have RNA expression testing, which is a great new gift for cancer treatment. It's still really important to have tumor DNA sequencing. It tells us how the genes are mutated and, what targeted therapies could benefit a person.
You're going to tell us a few other things that tumor DNA sequencing tells us that's really important that you really right now can't get from any other type of testing.
Yes.
Yeah.
Yeah. So here's the slide I covered early. Chemotherapy kills all rapidly reproducing cells, regardless of whether they are cancer cells or healthy stem cells.
I would think.
And targeted therapy works by targeting only the cancer cells that have the mutations. And that's the key to precision oncology. So the important thing is since we've been able to transcribe the human genome back in like 2005 or so, all of the fundamental approaches to dealing with cancer have been identifying specific mutations that occur at a high percentage in certain types of cancers, and then creating drugs that target just those mutations. The class of drugs are called tyrosine kinase inhibitors, typically, because they target a part of the gene that turns the gene on and off, called the kinase domain. A little more biology than you need to get into. But as a result of this, we now have access to hundreds, if not thousands, of drugs that target specific mutations based on DNA sequencing. So, once again, it's really important to have tumor DNA sequencing and know what your mutational profile is for a variety of reasons. And
So
we're gonna
just to
get
be
into
clear,
this.
because this can be really confusing and it is important, a targeted therapy is not chemotherapy.
Well, it's not, but it can be. And that's where the ADCs come in, which is targeted chemo. So, but we're not going to talk about those today. We're
No.
talking about tumor DNA
So
sequencing.
for our purposes to do with DNA testing,
Yeah,
when we're talking about targeted therapies, we are not talking about chemotherapy. It's a
correct.
different type of therapy altogether.
Yeah.
Just want to be clear about that.
Mhm. So, co-mutations. This is something that has largely been ignored by the medical system as a whole. And this is really sad because while I see a lot of oncologists embracing tumor DNA sequencing, they really miss the point. And what is important is it's not just the mutations you target with a drug, that can be targetable. It's really important to know what your co-mutations are. And these are mutations that may not have a drug that targets them, but can prevent or increase the benefit of a targeted therapy targeting another specific gene. So for example, you could have one mutation that is well targetable, there's a drug for it, but you have another mutation that prevents that drug from working, that is not even in the same gene. And so we call those co-mutations or inactivating mutations. And they're largely ignored for the most part right now. We spend a lot of our time really working on identifying co-mutations because they're well established. There's a lot of co-mutations that are significantly established. So we can look at, for example, we can look at a certain gene and a drug, and we can predict whether that person will have a 5-month response to the drug or a 25-month response to the drug just based on the co-mutations. So they really do modify how targeted therapies work. So takeaway, co-mutations are mutations that may or may not be targetable with drugs, but which can prevent or enhance the benefits of other targetable mutations. So knowing this information ensures you do not waste time on a targeted therapy that won't work for you, or you may require additional drugs to target those co-mutations. And one of the classic examples, which I won't get into, is EGFR positive lung cancers. There's all kinds of other mutations that can happen in the EGFR gene or other genes that can prevent the targetable therapies from working. So we also want to talk about spot panels. And spot panels, I think, are really the worst thing that's ever happened to tumor DNA sequencing. Spot panels are panels that look for key mutations. They will look for a small amount of mutations that have data on them. But since the data is constantly being updated and changing, these spot panels really don't do a lot of value to the medical system. They'll tell somebody that they have this mutation and that this drug targets it, but they won't give you the co-mutations. And so a lot of people will be told, oh, we have a drug that can target your mutation and you're going to do well. And then, 6 months later, the person's riddled with cancer because they thought they were going to have a good response, but the co-mutations required a different drug or extra drugs. Yeah. So spot panels, one of the worst things in my opinion. They really, they're deceptive. They're very deceptive.
Yeah, there's something about a kind of a lack of understanding present day for these to still be utilized and perceived as beneficial. But if we think about when most of the spot panels that are used in standard of care at this time came about, there really were only a handful of targeted therapies. There weren't thousands. And genetic testing was pretty much in its infancy since the Human Genome Project, 2003 officially, that came about or finished. So there was a time when kind of those spot panels, even if what you're saying I totally understand, is there's this element of so many different mutations you can test for and all these co-mutations. But I guess I just kind of want to kick a little love to the medical system here and the doctors that work within it and the barriers that they come up against, because there was a time when even the use of spot panels was very advanced, actually. Now, that
Mhm.
was well over a decade ago, and we're blown past that by far now, as we'll get into in time. But that's kind of partly the point, I think, of what Alex and his team do, which really is to keep up with the most current science and make sure the most current science is applied to every patient's care, and that doctors are educated as quickly as possible about what's possible.
Mhm.
So there was a time, I think, when these spot panels, they gave more information than we would have had otherwise.
Mhm.
But now I hear you. They do more harm than good because they make doctors think they're doing genetic testing when really they're not.
They're
And they make
not, no.
patients think that they know they're driving mutations and that they're on the right treatment when they rarely are from just these panels.
Yeah, yeah. And just to summarize, the spot panel will look at the key common mutations in the gene, whereas full gene panels will look at the entire length of the gene and show all of the mutations, whether they got targeted drugs or not. And I heard from an oncologist the other day, and I said, did you actually look at, do a proper DNA test? And he says, yeah, we did a fairly extensive one. It covered quite a few genes. And I said, well, how many mutations do you think there are in that gene? And we're looking at a very small gene. We're looking at the EGFR gene. And he says, oh, I've heard that there's at least, 10 to 15 different mutations in that gene. And, this panel looks at, a considerable amount of them. And so I had to educate him on that.
Okay, so
So
educate us. You're going to do this now?
I'm going to educate you on how many
All
different
right.
mutations can occur.
Excellent. I want to hear what you said.
Yeah. So aside from mutations, obviously, the 5 to 20 mutations they look at, or some spot panels will look at, 50 genes or something like that, but not the entire length of the gene. We want to know how many is many. And I'm going to show you right now how you can calculate how many different combinations of mutations you can get in a single
In
gene.
one gene.
One gene. So some basic math here. So to determine the possible combinations of mutations in a single gene, we need to take the power, or take 4. It's the 4 possible base pairs: adenine, thymine, cytosine, and guanine, designated by A, T, C, and G. And then we multiply them by the power of the length of the gene in base pairs. So remember, 3 base pairs are encode for 1 amino acid. And so the entire length of the gene, can be quite extensive. So for example, if we look at the largest human gene, it's called the dystrophin gene, it has 2.4 million base pairs. Now that's kind of exceptional. Many oncogenes have maybe a couple thousand base pairs, maybe 8,000, 10,000. Tumor suppressors are typically more complex genes because they have multiple functions and many different domains in them. And so they're usually much larger. But oncogenes are usually much smaller, and it's usually the oncogenes that we're looking at. So what you do is you take the 4 possible base pairs and you do it by the power of the length of the gene. So if we were to take a fraction of a gene, so let's say this dystrophin gene, we're just to take 150 base pairs. Remember, it's got 2.4 million. So if we were just to take 150 base pairs, then the way we would calculate that is we would take 4 by the power of 150. And so I put this into my calculator. I had to use a special calculator for this because, as you can see, the number is quite large. 4 by the power of 150. So this is the amount of possible combinations of mutations that happens in a tiny, tiny fraction of a gene. One gene, tiny fraction of it, barely the size of one exon, coding region. That number is significantly larger than all of the atoms based on hydrogen in the visible universe. When I say visible universe, I don't mean what we see with our eyes. I mean what we see with all of our technologies, every single satellite, everything. So this is way larger. So in a tiny, tiny fraction of one gene, you can have more combinations of mutations than you have atoms in the entire universe. That's fairly significant.
Okay, so the 11 or 20 that they were testing for
It's a
was
little off.
exhaustive. Got it. So
Slightly off.
yeah, that's a lot.
But
Yeah.
if you don't do the math, you'd never know.
Right. You'd think, wow, again, like I was just saying, well, it's better than we used to do. How could this not be good? But obviously the point is we want to be as thorough as we possibly can, and those co-mutations need to be identified as well. So what's driving the bus and what might get in the way of the treatment working needs to be identified. And the tumor DNA sequencing, if it's done right and it's a good panel, will help with that, is what you're getting at.
Totally. Yep, exactly. Yep. All of the atoms in the entire observable universe based on hydrogen, which is the basic fundamental atom element, is between 10 by the power of 78 and 10 by the power of 82. So remember, we have 22,000 genes. So, looking at every single possible mutation in one gene, I mean, I don't think we have enough paper on the planet to write those down.
Okay.
So luckily, we have these supercomputers these days that run through these trillions of mutations and tell us which ones are relevant and which ones are not.
So the point is we can assess for this.
Oh, yeah. Yeah.
Yeah,
We
right
can
now,
pick and choose what we want to see.
not science fiction. It's a real
No,
thing
it's
for
reality.
cancer
Yes.
patients.
Yes. So, takeaway. It's not possible to have a cancer without having any mutations. Just not possible. Every cancer is caused by DNA mutations and epigenetic alterations, which is different. We've talked about that in other videos. So in other words, if your doctor tells you that you do not have any markers for cancer, what they're actually saying is we've only used a very, very small panel, a spot panel, and we've only looked at a couple of mutations, and you didn't have some of the more common ones. So obviously that means you're going to be missing out on possible targeted options and co-mutations, knowing if that drug that is supposed to target your cancer is going to work or not. You can't know that without knowing your co-mutations. So proper tumor DNA testing. So now that you know all of that, how do you tell whether the test that you're getting, your DNA sequencing test, is going to be a good panel or a bad panel? And that's really important. Well, this is really important because for some people, they get this tiny little biopsy, and that's the only biopsy they get. They get one shot at doing it the right test. And that tumor sample gets used up regardless of whether it's a big panel or a small panel. And once that tumor sample is used up, there's no retesting. So, getting it right the first time with a nice large panel is essential because most people don't have the luxury of retesting, and it's expensive too. So the types of mutations that we're interested in, that a good tumor DNA test will identify.
And let me, just before you go here, Alex, let me just tell those of you watching at home, the reason why we're interested in this is because each of these pieces of data help us to dial in the perfect combination, or science don't like the word perfect, but the best possible combination of treatments for you. Each one of these variables makes a difference, and they need to all be considered.
And this is just a small, generalized list of types of mutations. It's actually many different types of mutations, but we won't get into that. So amplifications, basically, they're multiple copies of a gene. So typically, in theory, now, of course, everything's different in science, but in theory, you have, one copy of each gene, two versions of it, one from mom, one from dad. Of course, I'm really generalizing. You could get both copies from dad. You could get both copies from mom. Certain genes will have multiple copies. Like, for example, elephants have multiple copies of a gene called P53. And because of that, this P53 is a tumor suppressor gene. Because they have multiple copies of this gene, then they actually have much greater reduced risks of cancer, because this multiple copies of P53 can greatly reduce cancer. So amplifications. Now, if amplifications happen in a tumor DNA or in a oncogene, then it's a bad thing, because when that oncogene gets turned on, it can actually turn on all of the copies of the genes, and you get a massive amount of signaling from that oncogene. Now, you can also have deleted copies. Deleted copies typically happen in tumor suppressor genes in cancer. So we'll see amplifications of oncogenes, and then we'll see deletions in copies of tumor suppressor genes. And then you can also get single nucleotide changes. Now, we used to call them single nucleotide SNPs, but now we call them SNVs, variants. We just like to refer to them as single nucleotide changes. These are typically changes that will change the amino acid. So you change this one base pair, and it changes the entire amino acid that goes there and changes the function of the gene. And then you can also get variants of unknown significance. And single nucleotide changes, often the way they work is they will, if they happen in a tumor suppressor gene, they'll inactivate the gene. They'll change the structure of the gene. And so it can't bind onto its binding partners anymore. And in oncogenes, typically what happens is it does quite the opposite. It changes the shape of the gene. And so when the gene actually binds to its partner to get turned on just briefly, maybe to replace some tissues or something, it gets stuck to its partner and it gets stuck turned on. So kind of like the stuck brake pedal.
That's that constitutive
Constitutive activation is what we call it. Yeah. And then you have variants of unknown significance. Now, what these are mutations that happen in the genes that we're looking at. You know, let's say you have a panel of 550 genes, they're cancer-causing gene or cancer-associated genes. Variants of unknown significance are those mutations where they may be unique to you. So you may be the only person in the world that has this mutation in this gene at that point. Or they just don't have any data on them yet. You know, they haven't been tested, so we don't know if they make the gene more aggressive, less aggressive. You know, they could improve and make the cancers less aggressive. In fact, we actually had a case of an individual with a lung cancer, very rare situation. And when we sequenced his DNA, he had literally every single tumor suppressor and oncogene we saw was deleted. Just a massive amount of deletions. I've never seen so many deletions in this. It was a lung cancer. And, you know, the person had this tumor and they didn't get any treatment and the tumor started shrinking. And so, you know, the doctors are like, you know, what's going on? And so the patient thought, well, you know, I'm doing this and this and this, and I'm eating apricot seeds and I'm doing this. And that's why my tumor is shrinking. You know, I don't need treatment. And the reality is the cancer was so mutated, it couldn't sustain itself. And so it was literally just dying because it had too many mutations and couldn't sustain itself. So variants of unknown significance are these mutations that we don't know anything about, you know, that are potentially pathogenic, as we call them, or not. What we do is we do a fairly significant analysis of variants of unknown significance. We try to determine if they're pathogenic or not and whether they could be targetable or not. And the way we do that is we have about 10 different modeling software programs. Actually, there's about 20 of them we use now. And they model these genes and they use artificial intelligence. And then we also do something called sequence conservation, where we compare that gene to all of the other species that we can get a hold of, you know, mammals, you know, also primates and so on. And if there are no changes in those genes at that spot where the mutation is, then we know that that's highly conserved. And therefore, you know, evolution hasn't allowed any changes at that point. And so therefore, if you have a mutation at that point, and evolution hasn't allowed it, it's got to be pathogenic because
probably problematic.
Yeah, because evolution rules basically eradicates pathogenic genes over time. Once again, you want to have, and this is unfortunate that a lot of medical doctors are complaining about the panels and saying, I don't want to see all these variants of unknown significance in there. You know, I can't use them. You know, they're not important to me. Don't put them in there. And so a lot of tumor panels don't include them. And so you'd never know. And then so we get these cases where people hand these tumor DNA sequencing reports to us, and there's nothing on there. And that's because all they had was variants of unknown significance. You
So
know,
they need to have their testing done again with an eye.
Yeah. And so the doctors say, oh, there's no mutations. And clearly there's no mutations on the test. And because the panel didn't include the variants of unknown significance.
Because as we discussed earlier, you can't have cancer without having a number of mutations.
Exactly. And with all the amounts of different possible mutations, you know, we barely know anything about mutations compared to the amount of possible ones that could be there.
Well,
So
yeah, that big number that you showed us earlier, if
yeah,
every one of those can be in every gene, it's no surprising most of these tests don't come back with almost all VUSs.
yeah. Well, the computers weed out the ones that are insignificant. In other words, if they don't cause a change to the amino acid, we're not really interested in them for the most part. And I'm generalizing.
Yes.
There are actual mutations that don't change the amino acid that we're very interested in that are targetable. But we'll talk about that some other time. That's
Okay.
that's a little too complicated for this video.
All right.
But yes, in general, we're looking at mutations where the amino acids get changed. So if you do get a tumor DNA sequencing panel, make sure you get the VUSs included. Make sure it does include them. If it doesn't include them, don't do that panel. Don't use that panel.
Don't waste your
No,
biopsy
don't
sample.
waste your time. Yes. So there's also other things that are involved in determining, whether certain drugs will work. So one of these things is called microsatellite instability, or MSI high. And then there's also mismatch repair deficiency. Mismatch, both of these are alterations to DNA. I won't get into microsatellites. You can Google them, but basically microsatellites are just little pieces of repeat codons spread throughout the DNA that provide a fingerprint. They're used in forensics and paternity because our microsatellite fingerprint is going to be unique and is going to be able to be used to determine whether you're related to somebody or not. And that's why they're used for that purpose. However, if you have microsatellite unstable cancer, that means you accumulate a lot of variations and mutations. And what happens then is a class of drugs called immunotherapy, or PD-1 inhibitors, can definitely provide benefits. So patients with microsatellite instability, their tumors look very different than the normal cells. And so they're easier to recognize by the immune system. And because of that, if you use a drug or that it boosts the T cells, such as a PD-1 inhibitor, then you have a much better efficiency. In other words, it's going to be easier for the immune system to recognize and kill those tumor cells because they have microsatellite instability. And the same with mismatch repair deficiency. If you have mismatch repair deficiency, you can't repair DNA mutations, and they build up and they make the tumors look very different. There's also programmed death 1 expression. Importantly, this is something that tells you whether PD-1 inhibitors work. We used to be all focused on PD-1 expression. It's now included with many different panels. We really pushed for that to be included in the early days on certain panels. It tells you whether, now not all cancers is it relevant, but for example, in lung cancers, maybe triple negative breast cancers, in certain types of cancers, high PDL expression, PD-L1 expression tells you whether a class of immunotherapy drugs will work or not. And then there's high tumor mutation burden. And so if you have high tumor mutation burden, you have more than 10 mutations per million base pairs. And I'm going to go over each one of these. So yeah, basically, this is a system of DNA repair that stops working properly, results in an accumulation of mutations. And once again, it tells us whether these immune checkpoint inhibitors, called immune therapy, specifically PD-1 inhibitors, there's also CTLA4 inhibitors, tells whether these drugs will be really effective or not very effective. But once again, it's not the only factor that you could use to tell whether immune therapy is going to work or not. And then programmed death 1. This is a test that's commonly done when you get your tumor DNA sequencing because it's easy to do. If you have high PD-L1, then these drugs will also work for you depending on the type of cancer you have. If you do get PD-1 testing, make sure you get what's called the combined positive score. So they used to just test the tumor cells for the PD-1, but now what they do is they test all of the cells in the in the tumor microenvironment. So the cells that actually support and help the tumor, that are recruited by the tumor, like fibroblasts and different cells like that that are in the tumor microenvironment, they test them for PD-L1 too. And that's called the combined positive score. So if you do get a test that includes PD-L1 testing, and I suggest you do, then make sure it is a combined positive score. It will say CPS. High tumor mutation burden, this results in a lot of mutations, and it's basically qualified by how many mutations per million base pairs of DNA you have. So if you have 10 or more mutations per million base pairs, megabase, that's MB, then you have high TMB. And this lets us know whether immune checkpoint inhibitors will work. Now, once again, that works for a lot of solid tumors, but not all of them. So for example, certain types of colorectal cancers, if you have high TMB, immune therapy doesn't really work that well. So it really depends on the cancer. You want to know this information regardless of what type of cancer you have. But in general, the FDA has approved immune therapies for any tumor, any solid tumor that has high tumor mutation burden. In other words, if you have more than 10 mutations per million base pairs of DNA, then you could probably get immune therapy as an FDA-approved treatment. Okay, so here's the considerations of choosing a good sequencing panel. For the most accurate and beneficial treatment, you must ensure you get a thorough tumor DNA sequencing panel, not a spot panel, and not a germline or inherited gene panel. So make sure you know the difference. A tumor DNA sequencing is done specifically on the tumor cells. Now, there are also liquid tumor DNA sequencing panels. We're going to do another video about that. They are relatively new, and they definitely are a good solution. We're not going to talk about them today, though. Typically, germline or inherited gene testing is done on blood. So a lot of people confuse germline and inherited panels with these new liquid testing panels of tumor DNA. And once again, we're going to cover that in a different video. But in general, tumor DNA sequencing is best done on the tumor sample, and it's not typically done from blood in most cases. Also, ensure you use a panel that sequences the entire length of the gene, not just a spot panel. And when I say that, I mean, all coding areas of the gene, all the exons. And then also, you want to have a tumor DNA sequencing panel that covers a minimum of about 315 to 600 genes or so. More than that is a bit of overkill in this day and age with where things are at. But, typically 550 genes is a good panel. Okay, and then you also want to ensure that the panel provides you with the variants of unknown significance, as these are often actually very, very significant, and they get updated all the time. One day it might be called a variant of unknown significance. The next day it could be a targeted gene or targeted mutation. And then once again, ensure the panel includes tumor mutation burden testing, microsatellite and mismatch repair testing, and PD-1 CPS testing. That's my spiel.
All right, thank you, Alex. Gosh, I think I ended up with a whole bunch of questions, but you covered a whole lot of stuff there about what is most important for people to know. What I'm going to do for those of you watching this is I'm going to create a little PDF for you to download that has that short list, although probably some of you are taking notes, but it'll have the short list of the things that Alex wants you to make sure your tumor DNA sequencing panel has in it. And of course, Alex and his team are here to help coordinate testing for you. They'll track down your biopsy samples. They'll get your testing done. They'll sort it out with your doctor, and they'll help figure out what treatment that you need most. And if you're not sure if tumor DNA sequencing is right for you, or maybe you've had tumor DNA sequencing and you want to have those results analyzed properly, Alex and his team are here to help you understand what tests you really need. And they're definitely happy to take your existing genetic testing and do an analysis on that and make sure that every piece of data that can benefit you is being applied to your care now and down the road as and when needed. If you'd like, independent of any testing, or again, just to get the ball rolling, if you'd like to have a chat with Alex about your case specifically, we have a one-on-one 1-hour consultation. Alex and his team will gather your medical records, review, do some research about all possible options for you, and then meet with you for a whole hour and talk with you about everything they've discovered for your case and their best recommendations. You get a written report and a recording of this consultation so you can just be fully present and ask your questions and learn. And you've got a whole hour with these experts. I highly recommend it. We get lots of really great feedback. almost every single consultation that Alex and his team do results in some treatment recommendations that you wouldn't otherwise know of. So,
Yeah, and in many of these cases, we find that the patient does have good testing, but it just hasn't been looked at. And then other cases we find that they had the wrong tests.
And of course, visit our YouTube channel and subscribe. There's lots of new videos coming up all the time, certainly a couple every week on all sorts of things that are important for you to know and just stay on top of. And you're always welcome, of course, to reach out to us. You can visit the website at ctoam.com and just fill out the contact form and ask us your questions. It's our mission that you are as well informed as possible and that you know how to advocate for yourself with your doctor as effectively as possible and with minimal stress for you. We're all about this, figuring out what you need and how to help you get it. So let us know how we can be of assistance. I hope you found this informative, and please make sure you send us your feedback and any other questions that this video brought up for you. We'll be happy to answer them.